I'm Gavin Harder. I'm a chemistry major, captain of the football team, sort of shot put for the track and field team.
My name is Leah Walksmith. I am a biochem major and I pole vault here at Dickinson.
I'm Jason Gavinonis. I'm an assistant professor in the chemistry department. My students in lab here are working on neglected tropical diseases. I found that athletes at Dickinson do make really great research students. The parasites are all the same in that region. There's a very large overlap in the mindset between scientific research and athletics. It is very performance-based, it's very competitive. In this case, we're competing against nature. So a lot of times you hear of what they call the big three tropical diseases: HIV, malaria, tuberculosis. What we're looking at are a series of what are called neglected tropical diseases. And the two flavors of that you usually hear of are Chagas disease and African sleeping sickness. And so these are single-celled organisms that generally live in the guts of insects and affect you when you've been bitten by them. It could be up to 20, 30 years reduced life expectancy, serious cardiac complications. Basically, if the bug gets into the heart muscle, it can multiply and grow and have these cysts forming in your heart. So what we're looking at is new ways to treat these diseases that don't receive a lot of attention from the medical community.
So I focus directly with Chagas disease and the parasite that causes that. When the parasite infects the body, the immune system responds by releasing free radical oxidizers. And the enzyme that we're trying to isolate acts as like an antioxidant. This enzyme is a two-part enzyme. It only works when it's fit together. So I try to make the protein sequence that'll fit in between the enzyme and not allow it to bind together.
I looked at about 200 proteins that are in these protozoan parasites. We are looking at proteins that have two parts that come together to be functional, and that's the area that we will target to ultimately break apart the protein, which would then kill the parasite.
And these could be very promising leads for the development of new drugs for these neglected diseases of the third world.
We are able to work in a lab this closely with the professor and also have this much freedom. It's just a real good experience for the workplace after I graduate.
They're doing all of it. I'm just there to make sure that, if they stray too far off the path, that I could kind of point them in the right direction. And so that is something that I think is really unique about Dickinson, is that we do have the infrastructure in place for students to do summer research during the semester, and work on very real and relevant problems